I was just like, I know this.
The bus is crazy. It's Sammy. So like, I want to have every day. And then when I get home, I change my clothes.
So that means she likes the right? Only if it's better. Yeah. Exactly. Because yeah, you have to like everything. Frames you have? You're bored. Because I went to like over winter break, we went to like a Southeast Asian store. The frames are so cheap, and then like to get like those all like everything's so cheap. Like in Vietnam, these are $10. They're super cheap. Yeah. I got my Whoa, I know. Because I have one handmade. I have one that was like really expensive. But I go all the way back. And I used to like, I was like, I need to get cheap ones. Because I always fall asleep and they were like 300 something bucks. No, and then yes. Yeah.
Okay, does everybody have a copy of the lecture and lab? They got it on Tuesday. Alright, so I don't have the exams graded yet. I'll have them done over the weekend and post them, and you'll get them back in class on Tuesday. Okay? So I worked on them last night. I've got the Scantrons run and the first page of the village graded. So I've got a couple more pieces to go. As soon as I get those posted, I'll put them up online. Alright, so we're starting heart this unit. Your textbook actually starts out with the blood cells, and it's what many faculty start out the semester with. I put the blood cells after the heart. You don't need to know what the blood cells do to understand how the heart works and heart physiology and the blood vessels. And so I combine blood cells with the immune system, because the immune system is looking at the function of one of the blood cell types, the lymphocyte. So we combine that as a unit when we're done here. Now that we've covered embryology, well, not covered, I've introduced you to embryology, we'll look at most of our systems with a short preview of how the organ develops. All right, the heart is really complicated. As it turns and twists and cavities form inside the heart and different walls are added to it. And even with the computerized animation that we have, it's really hard to find one that's good. So, essentially, what you need to know is that the fetal heart is formed of two blood vessels, and this is in the diagrams in your lecture notes. Behind the ventral cavity are two aortas, dorsal aortas, that will fuse together. And this single tube is what develops into the heart. Okay? Now, if blood flow is in this direction, and we have the same continuing pattern here, we have blood flowing into the heart and out of the heart. All right? when we look at the heart, blood flows into the heart into what we'll talk about later today, into two chambers identified as receiving chambers. Right? Those are these two chambers right here. Want to know the name of the receiving chambers? Atria. Single is atrium, plural is atria. Atria rather. And then blood from there flows into thicker walled chambers, which will now pump the blood out. Alright? So blood is flowing in to the top and the bottom of the right atrium, and blood is flowing into the lateral aspects of the left atrium. In relationship to the rest of the heart, these are more superior than the ventricles are. But if we look at the pattern of blood flow here, the blood flow in what would become the receiving chamber is coming from underneath, and it's being pumped out the top, which still occurs with the heart because the top of the chambers are here, and the vessels leaving are the aorta and the pulmonary trunk. So what we have in this fusion is a migration where this lower portion comes around and up to the top as well. And that's kind of complicated to show. So let me just show you a quick video. It's not in any detail at all. This is actually of a frog heart. It doesn't last very long. So that is actually going to form a single fused vessel. These are two mesodermal structures. The heart develops from mesoderm. And we'll get a little closer up view here. See how it's migrating internally. We're seeing walls that are septiform as we have several different chambers. The frog heart actually has only one ventricle, but two atria. So you can see how it changed positions and moved around, and all of that's hard to capture. When students study embryology, they study as a series of slides, and so trying to put that into one vehicle can be difficult. So let's watch it one more time, then we'll look at some still images of human heart development. Those are the atria, blood flowing from the right to the left, and now the atria moving up and around to their normal position. So on the left would be the atria, and the larger chambers would be the ventricles. So what happens is a very similar process in humans occurs. It's just we have 4 chambers developing with the 2 atria and the 2 ventricles. So internally, there's a formation of the septum. So this diagram is in your lecture notes. And we have these 2 vessels forming a single tubular, and it's a little similar to what you saw with the frog embryo. Here's the ventricle. And then the atria down here are going to migrate up and around to become the atria of the complete heart. So this is kind of where we left our embryology on Thursday last week. Here would be the mesoderm and the endoderm and the ectoderm. This is our ventral cavity with the developing gut, still open to the yolk sac down here. The 2 blood vessels are seen right here. And then that would form the lumen of the heart, and the mesoderm surrounding it is going to form the cardiac muscle. Okay? And essentially, that's really what you need to, all you need to know. I want to look at some you don't need to know these different terms, but here are the 2 vessels coming in, all right, the 2 vessels leaving. And you can see it, these 2 vessels are going to form the receiving chambers of the heart, which are up on the top. You can kind of see them migrating up and around as the receiving chamber. So all of our vessels are associated, except for the inferior vena cava, with the base of the heart at the top due to that migration. Internally, we have to have this cavitation and the formation of the septa for the human, creating 2 atria and 2 ventricles. Now the 2 atria are easier because we have 2 vessels, each forming a receiving chamber, all right? But separating the ventricles, this single tube here, into separate structures requires the formation of the interventricular septi. And if that doesn't occur properly, we have a baby born with a hole in its septum. Now, it's not uncommon that most babies have, they should have an opening between the 2 atria. We'll look at that. It closes after birth when pressure changes in the heart, all right? But it's not normal to be born with a hole in the ventricular septum, allowing a mixture of blood between the right and the left sides. All right, so moving back over to this diagram here, in the frog heart that we saw, has a single ventricle and 2 atria. All right, so 2 vessels are emptying into the atria, and 2 vessels are leaving the ventricle, one going to the lungs and one going to the body. Same as we have, but they're coming out of the same vessel. Both of them are coming out of the same ventricle. In humans, we have 2 chambers, and the pump that pumps to the body is separate from the pump that pumps to the lung. This is okay in the fetus. A fetus could do just fine with a single ventricle, a single pump. Anybody know why? As soon as it's born, however, it's going to need 2 pumps. All right, so while it's a fetus, it's getting oxygen from its mother. And how does your crash? You're on the right track. Why does that make a difference? Okay, what causes blood to move? Pressure. All right, so pressure, blood moves from a site of high pressure to low pressure. All right, so the pump is creating the pressure. And with one pump, how many different pressures can we create? Just one. With 2 pumps, we can create 2 different pressures, all right? Because unless there's a disease, each pump is pumping the same volume, which is very important, all right? Because these circulations are interconnected, and they're not pumping the same volume, then we're going to have a problem somewhere as those volumes start backing up or catching up too fast. All right, so in a fish with gills, or some amphibian, or a fetus, what do these have in common as far as the site where oxygen is being exchanged? If this was the membrane where we had oxygen being exchanged, the gill surface and we had blood capillary blood on one side. In the fish or the amphibian, the source of the oxygen would be the river water or the sea water or wherever the oxygen was, right? So this would be the fluid that is carrying oxygen. So we would have pressure being created against that wall, the capillary wall or the gill wall, by 2 different fluids. Okay. What happens to this diagram? Well, where's the fluid in the fetus? Are we talking about the air sacs? Where is the fetus getting its oxygen? Where?
The umbilical cord?
Well, the umbilical cord is coming from what organ? Placenta. All right, so we're talking about fetal capillaries in the choriombili and the blood of the mother. All right, so both the sides of this membrane have a fluid force. In the fish, it's the oxygen in the water of the river or the ocean, okay, and the blood in the gills. What happens when this infant is born and the umbilical cord is cut? Where is the oxygen exchange now? We're still going to have capillary blood. Where's the oxygen exchange? In the alveoli of the lungs, and that is no longer fluid. So now what we have is alveolar air. Less pressure. Why does that make a difference? Does the fetus still have this situation of fluid on both sides of the tissue? Where would this occur in the newborn? Where would that occur in the newborn? What part of the body? You have it in you now. So on the right-hand side, capillary blood in the lungs, getting oxygen from the alveolar air sacs. And where's it taking that oxygen? Out to the tissues. And the capillaries are surrounded by tissue fluid from cells that need oxygen. So in systemic capillaries, those capillaries are going to have fluid on both sides of the capillary wall. In the pulmonary capillaries, they're going to have air on one side. Difference in pressure. Okay. So if we have just one pump, like the frog heart does. I did a paper on this for my A+P class my senior year in high school, and dissected snake hearts and frog hearts and fish hearts and embedded them in plastic. And you can buy those kits where you put coins or different things and it polymerizes and hardens. So I was illustrating the different chambers of the heart. And this is where I first started thinking about this. So in the fetus, before it's born, it really only needs to equilibrate the pressure here, which would be similar to the pressure in its systemic system. As soon as it takes a breath, and the alveolar sacs, which were filled with fluid, why do we need the alveolar sacs filled with fluid in the fetus if they're not being used? If they're not filled with fluid, what's going to happen? They're just going to grow solid. And when the baby takes a breath, there's no space there. So we have to have the sacs being developed, and the fluid helps them to keep from being solid. A few weeks before the baby is born, this fluid is absorbed, all right, especially if the baby is born vaginally. That pressure passing through the vaginal canal helps to squeeze extra fluid that hasn't been absorbed. The baby takes a breath, breaks all those hydrogen bonds, fills those air sacs with air, those alveolar sacs with air, and all of a sudden, pressure and resistance in the pulmonary system drops precipitously. Well, if the fetus had only one chamber, it's not going to grow a second ventricle really fast, so it has to be born with that second ventricle. What would be the problem if we pumped with one pressure now that we have two different pressure systems? With this higher pressure on this side and lower pressure on this side, water is going to get pushed into the alveolar air sacs. Pulmonary edema. That happens in congestive heart failure. When the left ventricle is weaker and blood backs up into the left atrium because it's not getting pumped out of the left ventricle. And then blood backs up into the vessels coming from the lungs and further until it reaches the capillaries of the lungs, and that higher pressure forces fluid into the alveolar air sacs, and that interferes with gas exchange. All right? So that would happen as soon as the baby was born if we didn't have this two-ventricle, two-pump system that pumps the same volume but different pressure. So a student this morning said, well, I don't understand how you can pump the same volume with two different pressures. And I said, okay, I've got two bottles, okay? One bottle is, very thin and flexible. One bottle is very thick and stiff. All it takes is a fingertip to push that one bottle and squeeze fluid out of it. The other one, I have to use the whole heel of my hand to get those walls to compress. So two different pressures, same volume coming out. Or you could imagine one bottle, same thickness of wall, but one bottle having a wide diameter and the other one having a very small diameter. Different resistance, okay? Different force that has to be applied and still get the same volume. Now, an infant isn't born with a thicker wall on the left side automatically because it's been pumping with the same force. All right, so it develops that thicker wall as it's being worked harder. So just like if you didn't use one arm, just kept it strapped to your body and only used the other arm or one leg, the actin and myosin protein inside the cells wouldn't be replaced, and the cells would become smaller, and your arm or your leg would become smaller. Okay? So if you worked one really, really hard and didn't work the other one really hard, the opposite, it would get bigger and the other one wouldn't. So the increase in thickness in the ventricular walls reflects the amount of work that each one ends up having to do over the period of your lifetime. Okay? So that's why we have two chambers coming in. We have blood coming from the lungs and blood coming from the body. And we have two chambers because we pump them out. Obviously, they have to overlap. Because if we had two separate chambers, the left ventricle pumping blood to the body, and it coming back to the left ventricle, to the left atrium, and the left ventricle would pump to the body, and it would come back to the left atrium. And the right ventricle pumped to the lungs, and it would come back to the right atrium. And then the right ventricle would pump to the lungs. We have two separate circulations. They wouldn't do your body any good. Your lungs would have lots of oxygen and be healthy, and the rest of your body would die. So we have an overlap, as you'll see today. All right? Blood from the right side of the heart is picked up from the body, and then going to the lungs, kind of like a figure eight, coming back to the heart, going to the body, coming back to the heart, going to the lungs, and we have an overlap. Okay? All right. Well, kind of a background of why we have this structure. Now let's look at the heart in location and some of the outer components of the heart. So in this particular unit, the anatomy portion of this course is the heart, the structures of the heart, the chambers of the heart, blood vessels, the names of the blood vessels in your body. That's the anatomy part. The physiology, which is what we're focused on today for the heart anyway, the physiology for the heart is muscle physiology. How does the heart muscle work? The ions and the neurotransmitters that cause the heart muscle to contract. And then the pressure cycles that cause the valves to open and close. Then we'll do blood vessels for anatomy, and then blood vessels contribute along with the heart to pressure. So we'll end up with blood pressure, and that's the physiology aspect. Okay? So the heart is located in a cavity. What do we call the two major cavities of the body? Do you remember that from 430? Well, major, ventral, okay? So the thoracic cavity is part of the ventral cavity, so that's our largest cavity here. And then the diaphragm separates the ventral cavity into abdominal and thoracic. So a subset is the thoracic cavity. And then the heart is in a subset of the thoracic cavity. What would that be? All right, if we look at a transverse section of the thoracic cavity, this would be the body wall with the muscle and skin and bones. And this is thoracic cavity, okay? So this would be the thoracic region of the ventral cavity. We'll put in the lungs. And then what cavity surrounds the lungs? Pleural cavity, all right? Notice I said surrounds, not within. It's a minor little thing, but it's a beef that I have with the textbooks that will say the heart is in the pericardial cavity or the lungs are in the pleural cavity, and they are not, okay? For instance, think of a down sleeping bag. All right, and well, let's change it. Let's think of this room. You're in this room. The walls of the room surround us, right? Are we in those walls? No. The walls have insulation in them, and we're surrounded by that, but we're not in it. Okay, so and you've probably seen this illustration before, if the lungs or the heart are growing into the ventral cavity, like a fist, and that's pushing against this fluid-filled sac, the edges of the sac attached to that fist, but I'm still not in that water-filled balloon. Okay, what's in the balloon? Serous fluid. So in the pleural cavity, the lungs are surrounded by the pleural cavity, but the only thing in the pleural cavity is serous fluid. Okay? So we'll draw that in here. So where the pleura is attached to the lung, remember it's called the visceral membrane. And then it's not attached here because this is where the bronchi and blood vessels are going to come in. So it's reflected off of that and attached to the posterior, lateral, anterior chest wall. And that is the pleural cavity. All right? And visceral pleura and parietal pleura are the two sets of membranes. And then we'll draw the same thing around the left lung. Put on the visceral pleura. Everything left is called the mediastinal cavity or the mediastinum. All right, so if I take the lungs out and I take away the pleura, everything that's left, or if I fill this all in, is mediastinal cavity. So this is mediastinal cavity. Above this, above the heart, is mediastinal cavity. Where the heart is mediastinal cavity. Between the heart and the sternum is mediastinal cavity. Even between the pleura and the chest wall, if I peel that out, there's a potential space there called the mediastinal cavity. Okay? So the heart is in the ventral cavity. It's in the thoracic cavity. And now the next space that the heart is in is the mediastinal cavity. So if I were to draw the heart in here, I'm going to make my pleura just a little bit smaller so I have room for more of my heart. have vessels coming out of my heart, even though this is supposed to be a transverse section. All right, so the heart is in the mediastinal cavity, and now it's going to be surrounded by its own membrane sac, which we know as the pericardium. All right, so it's going to be attached to the surface of the heart as the visceral pericardium, viscera meaning organ. All right, so this would be visceral pericardium. And then up where the vessels are found, it's going to reflect off of those vessels and form the parietal pericardium. And the space between is the pericardial cavity. So the heart is surrounded by the pericardial cavity, but is not inside it. I think the problem comes from people think of it more like a down sleeping bag. And they think of the cavity as being inside the sleeping bag, so you get inside the sleeping bag and you're inside the sleeping bag. But you're not in the space where the down and the air and the feathers all are, okay? So you're surrounded by the sleeping bag, but you're not actually between the layers of the walls that form that sleeping bag. Now, what would I have to be talking about if I wanted to include pericardial cavity in my list up here? This list is all the structures that, all the spaces that you could say the heart is within. If I wanted to include pericardial cavity up there, what would I have to say? What's inside the pericardial cavity? Same thing that's inside the serous. I'm just skipping the answer. There it is. Same thing as inside the pleural cavity. All right, so if I were to start off with serous fluid, then I would have ventral cavity, thoracic cavity, mediastinal cavity, or mediastinum is another way of doing it. Then I can include pericardial cavity. Serous fluid would be in the pericardial cavity, which is in the mediastinal cavity, which is in the thoracic, and so on. Okay? So again, everything that's not lung or pleural cavity is mediastinum. The heart's in the middle mediastinal cavity. The superior mediastinal cavity would contain the great vessels. If it was a child, part of the thymus, the part of the thymus would also be anterior. So anterior would be between the sternum and the heart. Posterior would have the esophagus, so esophagus would be in superior as well. So anything that's behind the heart, primarily the esophagus and sympathetic chain. Above the heart would be the vessels, the esophagus, the trachea. So anything that would be from the thoracic inlet to the top of the heart. Okay? So before we look further at the relationship of the heart to the lungs, let's take a closer look at the pericardium. Okay? As a serous membrane. Hi, kiddo. Hey. You want to keys to my office? Yeah. This is my son. Hi. Just broke down from college, so. Didn't expect you till this evening. We're all supposed to be at games.
Huh?
Do you know what schedule?
Yeah, I got it.
When do the games start?
2 and 5.
Okay. You're going to go there now, or?
I'm going to get lunch and go. That's okay.
Okay, but what do you need with the car?
Huh?
From my office? What do you want keys for?
I was going to get money for lunch, if that's okay.
From my office? Wallet's in the car.
What's in the car? Okay.
So there.
Thank you.
All right, so if we're talking about the visceral flora, it's just a serous membrane. And what's our definition of a serous membrane? That's bio 430 stuff. So we have 4 kinds of membranes: serous, mucus, synovial, and cutaneous, which is skin. So all of them but synovial, our definition is an epithelium plus connective tissue. So with skin, the epithelium is non-cornified, stratified squamous epithelium. And the connective tissue with every epithelium is loose, irregular, because it needs it to supply nutrients with the loose arrangement of fibers and lots of fluid. All right, so the papillary layer of our stratified squamous epithelium of skin, the epithelium lining your mouth, lining the uterus, lining the oviduct, lining the vas deferens is considered a mucous membrane. About all the different epithelia that we have are classified as the mucous membrane epithelium still, plus loose connective tissue. And then we have serous membranes. With serous membranes, it's always a simple squamous epithelium and a loose connective tissue. All right. So if we look at this image here of open heart surgery, here's the heart, and here's part of a lung. Notice how shiny that is? That's due to the serous fluid made by those simple squamous epithelial cells. So that's the reason why it's called a serous membrane. Our mucous membranes don't fit because they don't always make mucus. But this thin epithelium secretes a watery fluid for the purpose of lubrication. So for your lungs, as you breathe in and out, if that fluid isn't there, it hurts. It's called pleurisy. Okay? And so that helps to lubricate that movement. Now, we have the same similar type of membrane with the heart, but we add a couple of extra layers of tissue. So let's go back to this diagram. And this diagram is showing a view right up here where the atria, the receiving chamber, is merging with the blood vessel coming out of the heart. So I'm going to draw this diagram right here. You have it in your lecture notes, I believe.